Here we are to finish up Chapter 16. This last portion is very short. Concept 16.3 says a chromosome consists of a DNA molecule packed together with proteins. And although throughout this chapter we've kind of compared humans and E. coli bacteria, because it doesn't matter if you're talking about the prokaryotic bacteria DNA or you're talking about eukaryotic human cell DNA, there's a lot of similarities between how they replicate. But of course there are some differences. Human beings and other eukaryotes are not E. coli bacteria. So we're going to talk about some of the differences in the amount of DNA and the structure of the DNA. And we're going to talk specifically about what a chromosome is and what it looks like and how it consists of a DNA molecule kind of packed in a certain way or put together in a certain way with the proteins. And the bacterial chromosome, we already talked about, is a double-stranded circular DNA molecule with a very small amount of protein sort of embedded within. And that is different from our DNA. Our DNA, the eukaryotic chromosome, has linear DNA, and these linear DNA molecules are associated with a large amount of proteins. In a bacterium, the DNA is what we call supercoiled, and it's found in a region of the cell called the nucleoid. Remember, in a bacteria, we're talking about a prokaryotic cell, so it doesn't have a nucleus, it doesn't have the organelles. So it doesn't have a nucleus with a nuclear membrane, and it's not separated from the rest of the material, but the DNA is kind of coiled together in an area called the nucleoid of the cell. That's a little bit different than us, obviously, because our cells have a true nucleus and our DNA material is separated from the rest of the cell. So let's take a look at our DNA material and how it's kind of put together and what we see underneath the microscope. First, we're going to talk about something called chromatin. Chromatin is basically the DNA and all the proteins associated with the DNA. So chromatin is a complex of DNA and protein found in the nucleus of eukaryotic cells. Chromosomes fit into the nucleus through a very elaborate packing system, basically. You're talking about a very large amount of genetic information, and if you were to string the DNA out in one long linear thread, it's no way would it fit inside the nucleus of the cells. Human cells contain an enormous amount of genetic material in one single chromosome, and remember you have 46 of them, 23 pair. One single chromosome, if you stretched it out, would be thousands of times longer than the diameter of the cell nucleus. So you wouldn't think that there's any way it can fit inside there, but it's packed pretty well and pretty tightly, sort of in an origami sort of fashion to make it all fit. Here's a nice little animation that talks about the different levels of chromosome packing. Oh, got a problem there. So take a look at this animation. You can right-click and hit play, and you'll notice it talks about the way that the DNA material is so tightly packed that it's not all available for transcription. Transcription is the process that we're going to study in the next chapter, where sections of the DNA are read, and those instructions are then sent off to the ribosomes to make various proteins to do whatever the cell is that it needs to do. So because of the way this DNA is packed together, sometimes it's so tightly packed that certain sections can't be read, and it has to uncoil to allow the reading process and the transcription process. This would be like if you were going to Europe for a week. Whoops, hang on just a minute. I have to fix pop-up errors that keep popping up. This would be like if you were going to Europe for a week and you were packing, 7 or 8 days worth of clothing, and you had to pack them in such a tight, compact way that they would all fit. If you were to be asked at the airport to produce one particular shirt and you knew it was packed in the middle, you might not be able to get to it. So in a way, some of the DNA, the way it's packed together, it's not always available to be read or to go through the transcription process. So the way it's put together can somehow affect how the different DNA information is used and what proteins are made. And we're going to talk more about the transcription process actually in the next chapter. So let's take a look at some photography, some microscopes, and some illustration. So here we have some great illustration, and I highly suggest you look on page 320 and 321 because there's a great big 2-page diagram with lots of good information about the structure of the DNA and how it's packed in certain formats. If you're looking at your textbook, we're kind of starting on page 320 and working from left to right. So what we're starting with is the lowest level of packing or the lowest level of structure. We're going to kind of work our way up from that. So at the most basic level, DNA or chromosome is a long strand of DNA. So if we were to totally unwind it and unpack it, you would end up with this long strand, double helix shape of the DNA itself. So here we have the basic material, the DNA molecule. Now that DNA molecule is not just in one long thin strand. We know it's packed tightly together, and it's wrapped around these particular proteins that are called histones. So histones are these particular proteins that the DNA wraps twice tightly around, and they have this unique shape where they kind of have what they call kind of like a head and a tail, and the heads are packed toward the middle with the tails kind of pointing out toward the outside. And the DNA wraps tightly around these histone proteins and forms this little ball shape here. These little beads or balls are called nucleosomes, and they look very much in a microscope photo as beads on a string, or like perhaps a set of pearls on a necklace here. So these beads on a string are what we call the nucleosomes. That will be the histone proteins, and with the DNA wrapped around those histone proteins. Much of these histone proteins are made up of amino acids with a positive charge, and the DNA overall has a negative charge, so they are attracted to one another and they're fairly tightly bound. This basically what you see, this bead on a string, again, the beads are the histone complexes themselves with the DNA wrapped around, and the material between is called linker DNA. What looks just like the thin strand of string here is called linker DNA. Now that is going to be accumulated or packed together in a larger form, which you can start to see off to the right here. So there's another histone protein called H1 that helps in this whole packing process, and we'll look at that next. So on the next slide, this will be off to the page 321. We have what we call these fibers, and this is basically those linker DNA and the histone bundles. They begin to pack onto one another, and they fold over one another until they form these fibers. And you can see these fibers in these TEM tunneling electron microscope photos here. Those fibers then in turn begin to form loops, and you can see the loop formation here, and then again in your textbook, and in this TEM right here. This loop formation further compresses or packs the DNA, and then those looped domains is what they're called, eventually form what we see when we look in the microscope at a karyotype. So in a karyotype, we may be looking at this. So we have the two chromatids here that make up the chromosome, or the replicated chromosome. Each one of these is a chromosome. When they're connected together, they're called the two chromatids. So in metaphase, when they are at their most compact, when they're at their most pushed together, they're easiest to see, and it would look just like this right here. And this would be where we take our karyotype picture, and then we can line up all of the chromosomes side by side, and we can look at all 23 pair. But notice the amount of packing that goes on. So if we start to unravel this set of chromatids, this one chromosome and the chromosome pair here, first we get these fibers, these loop domains, and then we can start to pull them apart, and then we would get to the various nucleosomes, and then further pull that apart all the way down to the thin strand of DNA. So to fit all that genetic material inside the nucleus, it takes some pretty ingenious folding and bending and looping and packing to get it to fit. So going back to this picture here, so this would be during metaphase of cell division. The chromatin material is not always packed this tightly. Obviously, we know that if we look at, say, interphase, we know that we won't see the chromatin packed together in this shape quite as tightly. But the chromatin kind of undergoes various changes throughout the whole cell cycle. So in interphase, it's a bit more spread out, and it might be organized into 10 nanometer fibers, and then more compacted into 30 nanometer size fibers through different folding and looping processes. And I already mentioned that because of the way it's folded and compacted, not all of the DNA is exposed and available for transcription at different times. So the DNA will unfold and unloop and unpack and repack at various times throughout the entire cell cycle. And at one time, in interphase, you can't really see the chromosomes as clearly because they're more stretched out and they're not quite as compacted. And at one time it was thought that all of the chromosomes in an interphase were just kind of all thrown together and intermingled together, but that's not really the case. They're still fairly organized, and it used to be thought that they were kind of like a big bowl of spaghetti, to where it was just a tangled mass inside the nucleus of all these chromosomes, and then somehow they kind of all separated and then compacted to form what we see in metaphase. But that's not really true, and we'll take a look at a slide that kind of shows how it's not just a mixed bowl of spaghetti noodles. Even in interphase, when they're not highly compacted, there's still some structure to the whole layout of the DNA molecules. So in this cool photograph, using some various microscopy techniques, what they've done is they've marked the chromosomes using a particular coloration of some chemicals or dyes to colorize the particular chromosomes. So they've marked them in interphase, and note down here, this is interphase. So this is when the chromosomes are not highly compacted and they're more diffuse. But notice, you'll see that they are not just completely all intermixed. So this would be like after they start to go through metaphase and they are fully compacted, and we could take a photograph and lay out a karyotype like what you see here. But here's an interphase. Notice it's not just like a big bowl of spaghetti. How you have the chromosomes, even though they're not completely compacted, they kind of have their own little separate areas and they're not all intermixed. And also notice that the homologous pairs are not necessarily located together. So here you kind of have this turquoise color, and the homologous pair turquoise is kind of over here. Or here you have this kind of purple color, and then the other purple one is right over here. So even though they do compress and then sort of align themselves together during the whole mitosis, cell division, they're not necessarily lined up together, and they're not all intermixed in interphase. So this is kind of a cool shot. Even when they are more spread out, they kind of have some structure to them, and it's not just a random pile of chromosomes inside the nucleus of the cell. The cool thing to think about too is the way they do all of this folding that we just showed and talked about. I'll go back to the slide previous here. So you have this entire folding process to get to this particular shape that we see. Well, think about how precise that folding has to be, because remember, say if there's a gene right here, well, the other copy of that gene, remember the other allele will be in the same spot on the other homologous chromosome. And think about how precise the folding has to be to put that section of DNA at the same spot on both of them. So that's kind of amazing to think how complicated this whole folding process is, and I'm sure it's controlled by the chemical interaction of the various molecules that make up the DNA and the proteins here. So the last thing we want to say here is that most of the chromatin is fairly loosely packed inside the nucleus during interphase, and then of course it condenses prior to mitosis. That loosely packed chromatin is called euchromatin, and then during interphase, a few regions of the chromatin are highly condensed into what we call heterochromatin. And it's these areas that are tightly packed that are sometimes a problem, because the dense packing can make it difficult for the cell to get to certain pieces of the DNA to express certain genetic information. So if we look back at this picture here, all the loosely packed chromosome material is called euchromatin. And then even within this area of interphase where it's fairly loosely packed, there'll be some areas that are more tightly packed than others, and that's called heterochromatin. Remember, hetero means unlike or different. So you have a few regions of the chromatin, the centromeres and the telomeres, that are highly condensed. So not the entire piece of DNA is kind of spread out. Some parts are still fairly compacted together, and it's those areas that can be problematic that tend to have to fold and unfold if the cell is to get at that genetic information. And the histones, remember, are the proteins that the DNA wraps around. They can undergo different chemical modifications that can change how the chromatin is packed and looped and sorted together. So it's fairly complex. I mean, it's complex all the way from the chemical nature of what the DNA is made out of, and it's complex to the 3D structure of the DNA, and then it's complex to how it's all packed together to form a chromosome. So it's a lot of information, and there's a lot of organization at a whole variety of levels. So now that we know a bit about the DNA, the next piece of the puzzle is, well, what does the DNA do for the cell? So now we know how the DNA is replicated for the daughter cells, but that really doesn't do us any good if we don't use that information. That DNA is basically, remember, a code of information that tells the cells what proteins to make and what to do. So now we need to talk a little bit about gene expression. So how is that information transmitted into action, basically turning that DNA, turning that instruction into actual proteins? And that's the next chapter we're going to look at. So we've looked at transcription, which is creating copies of, we've looked at replication, which is creating copies. And in the next chapter, we're going to look at transcription and translation, which is taking that DNA and getting the instructions out to the protein manufacturing parts of the cell and then creating proteins. So in this chapter 16, we talked about DNA and what DNA is made out of, what it looks like, and how it replicates. And in the next chapter, we're going to look at how that information is used for transcription and translation and making it into proteins. We're going to stop right there. Hope you guys had a great Thanksgiving, and we're going to push on through for the last month. Like I said, we have about 2 or 3 more chapters to go, and we'll knock out this first semester of biology. You guys have been working really hard. I've been proud of many of you for your excellent grades and how hard you've been working and contacting me and doing your best to keep up with this online course. I know it's been quite the challenge, but I am looking forward to finishing up strong, and I'm looking forward to an even better semester coming up after the Christmas break. So give me a ring on the Schoology site or an email if you need anything from me. Otherwise, I highly suggest that you go over these notes and you go over your text and you look at this in multiple ways. Go check out the crash course videos and the other videos that I've uploaded. Use the various internet sites that are available, such as the various flip card vocabulary sites and that type of thing. And then be sure to contact me if you have any questions or need further help. Talk to you soon. Bye-bye.